Thank you very much, Jean, and thank you for the invitation. It's a real pleasure to be here and to have the occasion to show you a bit what we are doing in Bordeaux, but also to exchange with you and hopefully collaborate to further develop some applications. So first of all, I would like to introduce myself. I am a paleoanthropologist, meaning that I'm studying human evolution. So my main interest is the period between our early origins and the emergence of our species Homo sapiens. So roughly from 2.5 million years ago to 500,000 years ago or 300,000 years ago. So today I will mostly speak about the application of deformation approaches like deformetrica. I will also show a few other methods, but this is the one I have been using a lot recently, and that showed quite important results for our understanding of human evolution. And I would like to start. I won't give you a course on human evolution, but just to give you some introduction and some basics. How was the hominin fossil record in 1950? So what we knew at that time was very limited. There was Homo sapiens, our own species, of course, in red, but also a few other species like Neanderthals and Homo erectus, and only two very ancient hominins, Paranthropus here and Australopithecus in yellow, were known, with only a single species for each genus. So our view of human evolution was quite linear and like a cactus tree with very few branches. Nowadays, it's more similar to that. We know a lot of species, and we also know more about our deep time evolution up until maybe 8 to 9 million years ago. There are still a lot of questions regarding the relationships between all of these species, but we start to have a quite good understanding of the diversity and also of the ecology and the evolution of these species. But the main question is, how do we link them together and, how they are related? So it's a kind of puzzle that we need to solve. And thanks to the morphometric approaches, the geometric morphometric approaches that were developed in the last 20 years or so, there were some new opportunities to really understand and to characterize in a quantitative way how to link all of these species together. So I will show you some applications I've been focusing on, and especially on a few craniodental structures. One is the inner ear. So the inner ear is a very complex structure with, as you can see, a part of it is a kind of snail, the cochlea, and the other part are the semicircular canals, which are used for the orientation and the equilibrium. And this work, I do it in collaboration with Alessandro Scioli, that maybe some of you know. And we have been trying to understand independently the signal, especially the phylogenetic signal, between the semicircular canals and the cochlea. So as I said, the cochlea can be separated, and the cochlea is mostly related to audition, to hearing. And depending on the size, volume, and also the length and the number of parameters, this can influence the way we hear sounds. And here you have a plot where you can see, for example, early hominins, Homo erectus, and earlier species of hominins. And you can see that more or less they are in the range of Neanderthals and modern humans, which are here. And here is Australopithecus, and some specimens were quite similar to other specimens of Australopithecus. So there was some variation in the hearing capacities of low frequencies and high frequencies sounds for the different hominin species that existed.
Okay. To be clear, for me, whenever I saw a cochlea, I assumed it was a soft tissue, but you told me at lunch, actually, it's like a tunnel in the bone.
Yes, it is.
Okay.
It is. So, yes, it's actually what you see here is the imprint of inside the bone. And we don't have the soft tissues that are inside. We know from the living apes and living humans that there is a channel with some microstructures like cilia and so on. But we don't know the exact morphology for fossils. The only thing we know is the external morphology of this snail.
Okay.
Which is also something Alessandro, my colleague, is working on, trying to do some deformation of chimpanzees and humans, living ones, onto Australopithecus and earlier species to try to see which one would be the best, the best fit for these early hominins. And the semicircular canals, they are involved in the way we move and the way we move our head, especially in the equilibrium. So we have 3 canals oriented in the 3 directions to have the 3 dimension mobility. And we found, thanks to this analysis, that the semicircular canal are actually very helpful for understanding the phylogenetic relationships, so the relation between species. So it's not very clear here, but these are all extant primates that we could analyze, 25 genera, so it's quite a lot. And you can see the different groups, which are known based on DNA. And we can actually separate them in a quite similar way based on morphology. So this morphology of the semicircular canals that you can see here along the plot, you see the extreme shapes from one shape to another, based on depending on the area of the plot where you are. This morphology is useful to reconstruct the phylogeny and to understand how these species are related. And this is, for example, the phylomorphospace. So it's a combination of both the molecular signal and the morphology. And we can see that basically what we see with the DNA is reflected in the morphology. And we could also plot some fossils for which we don't have DNA and try to understand how they are linked to the present species. And for example, you see that Australopithecus is closest to humans, and it's far from the other apes that we know today. And here, these are some indications of the phylogenetic signal. So the closer to 1, the strongest it is. And you can see that it even reaches 1 here, and here it's exceeding 1. So it means that there is a very strong phylogenetic signal. And it could be used in replacement of DNA when we don't have DNA for very ancient primates fossils. And there is another structure, the one I'm working on, that is also very important, and it could show the same kind of signal. It's called the enamel-dentin junction. So basically, when you have a tooth, you have the crown, that is the part that is in your mouth, and this part is the enamel. If you remove the enamel, the white part, below, you have this morphology, which is the enamel-dentin junction, or EDJ. And why this part is important? Because most of the time it's not affected by wear. When you eat, you will wear the teeth, but this part is under the enamel, so it's protected. And if you remove it virtually with using scans, like with X-rays or other methods, you can access to this morphology. And this morphology is also very complex, more complex than the outer morphology, and it preserves more information. So for example, you can see here, the red part is the enamel, so the outer part in the crown, in the mouth. And below, in yellow, you have the enamel-dentin junction. And you can see that there are a lot of small crests and grooves and things that you cannot see outside. And these are very important to characterize the species. So for example, using deformation analysis, we can recognize and separate Homo erectus from Neanderthals and modern humans. And we can also link some other specimens that were found in Asia and are dating to maybe 500,000 years to 100,000 years to Homo erectus. But there was an assumption for years, since 2008, you can see this citation, that this morphology of the dentin was useful for taxonomy, but also for phylogeny However, it was never tested so far.
How much data, how much tip you have like this to have enough data to know that it's important to have this small like characteristic shapes?
Now we have hundreds of teeth for hominins, so we have quite a large sample. And it's also much larger than with DNA because DNA is destructive. When you want to extract DNA from a fossil, you need to break a piece and destroy it. So the number of fossils that you can analyze for DNA is very limited, very restricted. But with this approach, you can just do an X-ray scan. It's not destructive, and you can analyze hundreds of fossils. So basically, we can have a lot of information, much more than with DNA. And also, DNA is not preserved for very ancient periods of time, but this is preserved. So.
And could you tell us maybe, because when I see this yellow shape, to me it seems very random, a bit like a fingerprint, but you're actually saying that you have parts which are clearly reproducible for all Homo sapiens. And so, like, if you take this one, could you tell us maybe what are the structures that are going to be the same for everyone in the room? And,
sure.
And what are going to be the parts which actually is a fingerprint?
Yes. So all humans, they share the same kind of peaks. So you have 4 main peaks. Sometimes you have a 5th one here, and then there are a number of differences. So this is an accessory peak, something that is not present in everyone. And this kind of crest is very variable. The pattern is quite variable. But what is very important here, you have like a small shelf, a small depression here. And the crown is quite short. It's not very tall. And this is typical of Homo erectus. In modern humans, the crown is much taller. And the peaks are a bit higher too. And you have also a more simplified morphology. It's like a square, basically, with just the 4 peaks. So the reason is because we evolved toward a smaller jaw in reduction with the face during evolution. So we have smaller teeth, and our teeth are much more simple than the ones of our ancestors in Homo erectus.
Okay, so that's really interesting because I think when I see this shape, my eye was attracted to like the ridges and everything, but actually, so no, okay, this depends a lot, but you really dragged our attention to the boundary and the shape of the boundary.
Exactly. So what matters is the shape of these ridges here, and also the height and the position of these peaks. But it's true that these small ridges can matter because Homo erectus has a lot of digitations, a lot of ridges, while modern humans, they have a much more smooth surface. So that also matters, but let's say that in general, the main morphological changes that you can see are the main position of these peaks and the global configuration.
Maybe just
sorry.
Another question on this topic. Is there any functional like advantage or drawback to have some taller peaks, taller crown, and so on that makes this part to be preserved between different specimens of the same species? Or
in absolute, yes, I would say. But the thing is, we have very small teeth, so they are very compact. So normally, if you have tall crowns, they may be more sensitive to constraints and more likely to break if they are when they are short, they are more compact. But there are other things to take into account. The enamel thickness on top of the dentin, and enamel is very thick in hominins. So it compensates for variations in size and shape and proportions. And also hominins, where we are all omnivores, all the species that existed before us and including us, we are all omnivorous individuals. So we eat a bit of everything, but we don't wear much our teeth. It's not like some species that existed in parallel to us that were constantly eating, and they were wearing a lot their teeth, or some were eating very hard food, and the teeth were more susceptible to they were more they could break more easily. So there are a lot of parameters to take into account. It's not just the shape, but also the size and the kind of diet, and also enamel thickness and so on. And this is quite interesting because while the tooth morphology is also adapted to a specific diet, actually our morphology is more generalist morphology. Pigs also have similar molar morphology to humans. That's because we are omnivores and we don't need a very specific type of teeth to eat some kind of food. But what is interesting is that the ADJ, this part, is actually very conservative and is not changing very quickly for its evolution. So what changes is the external part, meaning that it's the enamel thickness that is changing very quickly. But below, it remains quite stable. And among a species, it takes a long time. And when a species evolves into another species, you can see changes at this level, but not if within a species there are some variations, it remains quite stable.
Okay. And you don't have access to the root shape?
Oh, yes, we do.
Okay.
We do. The roots are more complicated to study because they are not always homologous. Sometimes you have 1 root, 2 roots, 3 roots. It's also related to diet. So we know that some of our ancestors were probably eating a bit more hard food than us. They have very massive roots, but there is also genetics and a lot of things to take into account. So the roots are more difficult to study, but we also do study the roots. So as I was saying that there was this assumption that this part, the ADJ, is very informative for phylogeny, but I wanted to test it. So I used the same kind of sample that we use for the inner ear. So basically using a number of specimens for each species and genus. And the result was very similar to the one we got with the inner ear, with the same kind of group. So here you have the American monkeys. Here you have all the baboons and relative of the baboons, and here you have the great apes and lesser apes with Neanderthals falling near to humans, which makes sense and is quite nice. Orangutans are the farthest from our branch, and gorilla and chimpanzees are closer. So it all makes sense in terms of evolution compared with what we know from DNA. And we could also project other fossils. That this one, for example, Dryopithecus, is dating to 12 to 16 million years ago. And we can see that the morphology is compatible with something that is more primitive than orangutans. And we know that orangutans separated from the African apes, including humans, around 15 to 18 million years ago. So in terms of evolution, understanding of evolution, it all fits. And we have the same indices with very close values to 1. And some values are even exceeding 1, largely exceeding 1, up to 4. So it means that there is a very, very strong phylogenetic signal, and it can be, in fact, it can be used as reliably as DNA to estimate the relationships and to understand also if a species is, or a specimen belongs to a particular species.
Okay, so maybe you're going to discuss that later, but to be clear, like I don't know how you produce those pictures. So it's like you took the teeth, you took some measurements, you had a table.
Yes.
And then
I didn't want to go too much into details, but I can explain to you the method. So basically, I did geometric morphometric analysis using the deformetrical, using the deformation-based approach. Only on the first molars, so that's what I was rating here. So only the first molar at the moment. I plan to do it for all kind of teeth, but as a starter, I tried on the first molar. And we have 10 teeth for each genus of extant primates, and also for the 2 fossils that we included here. We do the geometric morphometric analysis, PCA or CVA or whatever statistical analysis. And then there are now some packages that can allow you to combine the molecular information with a molecular tree to the geometric morphometric analysis and to see if there is a correlation.
And how are they called?
Phylotool, and I can give you Geomorph Phylotools. There are different packages to do that. And once you do that, you can create this kind of it's called phylomorphospace. So the combination of phylogenetic DNA and morphology, and it calculates the distances and also shows the most likely relationships based on both informations. And in fact, you can see statistically if it fits or not. And in this case, it fits very well. If I know that there are some studies of the cranial morphology, for example, they have done the same for the external cranium. And the signal is roughly 50% correlation. Here we are at 99%. So some structures in the cranium or in the teeth are more suitable to understand the relationships between species than others.
Okay, and the correlation, it's the correlation between the genetic and phenotypic.
Yeah, exactly. So again, to try to solve this puzzle, and I'm working on specific topics. Here, my main question is, when did our genus, Homo, emerge? And we know in Africa, there are a lot of species and a lot of genera in this time period, in this time frame. So most of the colleagues assume now that our genus evolved sometime between 2.8 and 1.8 million. There are fossils that were described to be to belong to our genus, but we don't know the species, so they call Homo sp. And there are then other species that that have been described, like Homo habilis, Homo dolphensis, and Homo erectus. I don't want to go too much into details, but just to give you an idea that people have been recognizing a number of species, and some are not yet even described or characterized as a species, but humans are supposed to have evolved since 2.8 or 2.6 million years. And in parallel, there are other species of Australopithecus and Paranthropus. So this is what we assume to know at the moment. And this is based on the external morphology and on the studies that have been conducted since the 1930s on the large sample of African material. And one of the specimens was found in Malawi. So my first case was to go and investigate this specimen in Malawi. And this is this fragment of mandible. So you see it's not very well preserved, two pieces that you can put together. And some colleagues, when they described the fossil, they said, okay, it's probably the same as Homo rudolfensis. So this very square shape that is different from the more elongated shape that you can see on the side. However, when I started to look at it and I just superimposed the mandible on top of the others, I saw that if you take the proportions of the molars, they all fit with the more elongated shape. So probably it's more similar to what we call Homo habilis than to the other species. So that was the first, and I realized that it was a big mess and nobody really understands things. If you just look at them or if you try to take basic measurements, it never works, basically. The other problem is that this fossil is very mineralized, so X-rays don't work. If you do an X-ray, you see just all the same gray everywhere inside, and you don't see inside any structure. So we had to apply a new method that is called neutron microtomography. So basically, you need a nuclear reactor and a special facility that is related to this nuclear reactor with a neutron beam that will irradiate the fossil, and then there is a detector behind. And this detector will record kind of radiograph, and then you just make rotations of the fossil for 360 degrees, and you record each radiograph on the detector. And then you can get, like with X-rays, 3D volume, in which you can see inside, and you can see that here the enamel is darker, and then there is the dentin below. You see the roots and the bone here with the spongeous bone in the mandible. So it works with neutrons, and that was great. So we knew we could analyze the internal structure of this fossil, and especially the internal morphology of the teeth.
And also,
sure,
to ask you questions. So it's never clear to me at which stage do those bones stop being fossils? Because, okay, this is very old, so it's a fossil, but when we talk about a Neanderthal bone from 50,000 years ago, it's still real bone, or it's already calcified? Or,
It depends what you call real bone. There are still biomolecules inside, so there can be some collagen in Neanderthals, in Neanderthal bones. You can still find collagen, which is a large constituent of bone, and there is also a large majority of calcium, which is the main constituent of bone. But there is also part of the bone that has been remineralized or with the water in the soil that infiltrates. You can have other kind of mineral elements that are going inside and make it more like a rock in terms of solidity. But in fact, when you find some fossils, they really look like normal bone or fresh bone. For example, I talked about Paranthropus at the beginning, and I excavated some fossils in South Africa. When you find them, you have the impression they are just from yesterday.
Okay.
But they have 2 to 3 million years. So it can be very impressive. The preservation and the external preservation or the visual impression is very tricky to assess how much remineralization was going on.
Okay, but when you put it in an X-ray machine, you quickly understand that it's not. It's much more opaque.
So we could have gone to a synchrotron machine, which is a very large instrument that can also give a good information on the internal structure and with very high resolution. But we realized that even with synchrotron, in some cases, we couldn't see anything inside. Neutrons can, because they are not sensitive to the same aspects. X-rays, they are absorbed by heavy elements, while with neutron, you can cross easily all the heavy elements, but the light elements like water stop neutrons. That's why they need a water shield around nuclear reactors and so on. So it's a different way to analyze, but it works in the same way. And we could do the geometric morphometric analysis of the ADJ using the deformation approach. And what was very interesting is that this specimen falls closer to Australopithecus than to humans. So for maybe 20 years, everybody assumed that it's early human. But in fact, now we are pretty sure that it's probably a different kind of Australopithecus species that we didn't recognize so far. That may look a bit like a human just because it maybe evolved from the same ancestor, but it's very far in terms of morphology from what we know of humans, including early humans that are in this sample that date to 2 million years ago. And when you do the complete analysis of the ADJ, but also of enamel thickness and of the root morphology, for all of these specimens, you can see that overall it's more Australopithecus-like. Sometimes there are some intermediate features with Homo, but it's not clearly Homo. And in general, it's Australopithecus. So this means that we can now recognize other species of Australopithecus that may look externally a bit like Homo and that were completely dismissed for a long time or not recognized for a long time. So that's why these new methods are important. Now we'll bring you a bit southern in South Africa, which is called the cradle of humankind. So in Africa, there are 2 main areas for human evolution. Eastern Africa, which now I'm starting to investigate. I've been focusing mostly on Southern Africa so far. So that will be the main focus of my topic, of my presentation. And in South Africa, basically all of the genera I presented before, humans, Paranthropus and Australopithecus, were there nearly at the same time, and we can find them in many sites. So here you can see what archaeological or paleoanthropological site looks like in South Africa. So all of this is what was a cave before. The cave was completely eroded from the top, and the chamber was open. And all the sediment and all the fossils that were inside the chamber are now open, and we can just dig in and find fossils. So basically, everything that is here in the middle is the former cave that was closed before and that now we can investigate. And in this kind of caves, I just say that there are a lot of fossils. Actually, I think probably thousands of fossils of our ancestors were found and our closest relatives. So you can see here the collection rooms in various museums and universities in South Africa. In some cases, even nearly complete skeletons were found. And also children. Here you have a child of Australopithecus. Actually, it's the first child of Australopithecus ever found. It was in 1925. And complete crania also of Paranthropus and other species. And why I do study teeth? First of all, they are the most abundant. It's very rare to find a complete or subcomplete cranium, but teeth, there are hundreds or thousands of them. So it's very easy to study them also. And now with the new approaches, whether it's neutrons or X-rays, we can really extract a lot of information from the teeth. And here I have, I'm showing just a small sample of what people have been assuming is the earliest humans that can be found in South Africa. And first of all, the first thing that you can see, they have very different morphologies. So if you look at the top part, these are upper molars, maxillary molars, and these are lower molars. And just looking at the upper molars, you can see that this one, for example, is very different from the other ones. It's much more complex with a lot of crest and wrinkling. The external shape also is like bulbous with rounded corners. In this case, it's almost square and very simple morphology. So just visually, you can see that there are things that are unlikely to be the same species. But again, we need to confirm it statistically, or at least to have some quantitative evidence. And one of the specimens is quite important because this is the child, the cranium of a child that was completely crushed. So it's thick like this, like 2 centimeters, completely crushed, but the teeth are not crushed. They are in good condition. So it's almost impossible to say anything about the cranium, but at least we have the teeth. And using the teeth that were forming in the cranium at that time, especially the upper molars here, we could identify the species based on what I just showed you before. So if we look first at the comparison between the teeth of this child with Australopithecus, Paranthropus, and humans, Homo erectus and modern humans, first of all, you can see that we have very small teeth compared to the other species that existed in the past. And you can also see that the shape of the dentin is different in this individual compared with Australopithecus or compared with Paranthropus. It looks more similar to that one. It's a bit smaller, but more or less the shape is the same. And one of my first study was based on classic landmarks. And you can see that with landmarks, it's difficult to separate between the three genera. There is a lot of overlap. And this specimen was falling in the very blurred area between Homo and Australopithecus, which in a way makes sense because we are speaking about the earliest humans and they are supposedly evolved from Australopithecus. So they should share a lot of morphological features with Australopithecus.
And the question is, here basically you perform some kind of PCA, you display the first two modes.
Yep.
And the question is, in all of your previous slides also, the numbers for your two axes, you know, like 77 and 22, they sum up to 100.
Yep.
That seems, you think, probably it's higher dimension. So actually, I'd be interested to know, with respect to the total variance, how faithful is this visualization? Because then you interpret the precise location.
So I will be happy to discuss that with you, especially because there are a lot of debates in our field now on the use of these kind of approaches. I don't use PCA because that's the issue. If I use just a simple PCA, I have maybe 50-60% of the variance on the first two axes. So that's why I'm using either between group PCA or canonical variate analysis. And in this way, with only 3 groups, with 3 genera, I have only 2 axes with 100% of the variance on these 2 axes.
Okay.
So now I know that many people in my field, at least, they don't like this much these methods. But the problem with PCA is that it's also quite subjective on how many pieces you investigate, until when do you go, and which one do you take more into consideration, and so on. So it's an endless debate, but I would be happy to have your opinion. And if you have other methods to suggest, I would be also happy to discuss it. And after I learned that there are other methods and I started to work with Deformetrica, then that's what I got. So with deformation analysis, we can have much more information, because what we can capture with landmarks is just the main peaks and then maybe the ridges with semi-landmarks. But in these early hominins, there were a lot of features, like for example, here you have a small depression, or you have some crests that are not always present, things that are not always homologous that you cannot capture with landmarks. But these things can be useful to discriminate between genera or species. And you can see that when we use this approach, we separate very well the three genera. And this specimen that was in the overlapping area before is now very close to humans. So it gave me some useful information and on statistic probabilities that this one is very likely one of the earliest humans. And other specimens that were supposedly human, they fall either closest to Australopithecus or to Paranthropus. And now that I've been studying this material for years, it all makes sense. Because, for example, these specimens, they start with the same letters, they come from the same site. And in this site, so far, only Paranthropus was found. And only these 3 teeth or 4 teeth were attributed to humans. But in fact, it's very likely that only Paranthropus was covered in this time interval in this site. And the same here for Australopithecus. So it's a very interesting, and when you understand also the site formation and what happened, and also how much time is preserved in each site, you can have a good idea of what's happening and what's going on. So I won't detail, but I have done the same kind of analysis for many kind of teeth. And as you can see, for molars, it works pretty well in the sense that you separate the 3 genera, and the fossils that were projected fall generally in one of the 3 groups. For premolars, it's a bit more tricky because there is some overlap, or some fossils they fall really in the middle. So in some cases, we actually don't know if they are more likely to be Australopithecus, Paranthropus, or Homo. That's something that I still need to work on. But by the end of this study that was published 2 years ago, at least we could identify some specimens that are very likely to be human, and others that are very unlikely to be humans. And this one here that you can see at the bottom is part of a mandible. It's not just 2 teeth, but it's a mandible with several teeth. And other colleagues, after I published these papers, told me, well, yes, but you just studied a tiny part of the teeth, but what about the rest of the specimen? Because they were sure that this specimen was human, and they were convinced that because of the morphology of the mandible and other parts, it must be Homo. I said, okay, so I will investigate other parts. And as you can see, this mandible is a bit distorted, so this part is not exactly at the same level as the other part, so there is a bit of deformation, and I had first to reconstruct. Here you can see also that it's a bit tilted.
And to be clear, presumably it happened after the death of the
Yes,
specimen.
exactly. So this happened in the soil, in the sediment, with the pressure of the sediment and a number of processes that happened after the death, yes. So I had to reconstruct the mandible. It was quite easy in this case because there was no plastic deformation, it's just breakage. So I just had to do segmentation along the breakage and put them back together. So of course, like every reconstructions, you cannot be sure 100% that it's correct, but it's much easier than if you have to do retro deformation, in which case you are never sure of the original morphology. And once I've done that,
You're doing it by hand, the reconstruction?
No, we've assisted with landmarks. So you can do it by hand, but you can also use landmarks on the breakage along the section, and put the other landmarks and just make them fit. Okay. There are many ways to do it. Yeah, and I in this paper I think I did 2 or 3 different reconstructions, and you can see here 3. Actually, they fall very close to each other in the plots. And again, so in this case, I didn't analyze all the bone morphology, but mostly the dental position, for the simple reason that bone morphology for the mandible is very variable, and it depends on the age of the individual. If it's a very old individual, the bone will tend to be shorter, and there are a lot of things related to bone resorption and with the age of the individual. So it's more difficult to study the bone itself, but at least the position of the teeth should not change. And we can see that even if it's not within the range of Paranthropus, it's closest to Paranthropus. And also here you have below the shape of the symphysis. So this part here where there is the chin in humans, in our ancestors there was no chin; it was flat. And you can separate very well humans, but there is a lot of overlap between Paranthropus and Australopithecus, so it's more difficult to discriminate. But again, if we look at the geometric morphometric analysis of the ADJ, these specimens almost consistently fall within Paranthropus, and it differs a lot from other fossils that were supposedly also humans, like OH7 here and this one. And these are supposedly one of the earliest humans in Eastern Africa. But you can see that they fall inside Australopithecus, and they are far from the other early humans that we have for sure, we know are humans.
Okay, and if you remind us, so the colors, so the ground truth species, they come from DNA analysis, or so it's...
No. So when I say we know for sure these are humans, it's based on morphology, and it's because they share the same characteristic as later species of Homo. So for example, these are mostly Homo erectus ergaster, which you can find in Africa and in Asia. And this species has a lot of features in common with other species like Neanderthals, even us, so on. But the first humans, really the first ones, it's more difficult because they are still intermediate between Australopithecus and humans. And to be characterized as Homo, they should share features that you can see in later species of Homo. And people have been assuming that was the case for all of these specimens, but in fact, whether it's the teeth, the mandible, or even the cranium, and now I'm studying also the cranium, I cannot quantify any of these features within Homo. All of these specimens for all of the structures, whether it's cranium, teeth, or mandible, they always fall with Australopithecus. Visually, they look like Homo in some ways, but it's just visual impression. So that's also why it's not always easy to convince colleagues, because they have been looking at these fossils for 20, 30 years, and they are convinced of something, but they don't want to accept when we show quantitative analysis, which are, in this case, for example, I don't put landmarks, so I don't have even my own bias, but yeah, it's how it is.
But these specimens were attributed to Homo before?
Yes.
Based on what? Because they said visually, but they had some arguments, I guess.
That's exactly my question. I asked the same question to my colleagues. I've presented a work 2 years ago in the main conference for paleoanthropologists, and I asked them, okay, now I've showed you all of these results, and I've showed you that not based on my own view, but based on these quantitative results, these specimens probably don't belong to Homo. Now tell me which features, which diagnostic traits you consider as typical of Homo. Nobody answered.
Okay.
So I'm still waiting for the reply of my colleagues. But they are still angry and they still don't want to accept this. So that's something that is really funny. You know, we are scientists, but we also, because we love what we do, and we are also our own impressions, our own, our own feelings, and sometimes it's difficult to question what we think we know, and yeah. So basically, this specimen that I showed you before, it has several teeth and the mandible. So when I did all of these analyses, I found that it's probably similar to a Paranthropus or close to Paranthropus, but it's not the species that we already know. So it must be a different species that was named Paranthropus capensis. I named it this way because the specimen was attributed to Telanthropus capensis when it was discovered. And my colleagues don't agree with me that it's Paranthropus now, but I just want to show you this picture because I think even visually, for me, it's very clear that, first of all, it's not the same as this. So this is Paranthropus robustus, and this is another the new species that I created. And you can see that the morphology is very different. So when I said it's not Homo, some colleagues accepted it's not Homo, but they said it must be Paranthropus robustus, the other species that is known in South Africa at the same time. So it must be the same species. But again, the morphology is very, very different. And just visually, you can easily say that they are not the same, they have not the same size, not the same morphology, but even then, they don't accept it.
And just it cannot be attributed to like age variation or sex or
They come from the same sediments, from the same layer, so they have the same age exactly in the same site.
Yeah. No, I was thinking the age of the monkey, like the age.
No, because so these are the permanent, the adult molars, and once they are formed, they do not change.
Okay.
And you can see here there is the wisdom tooth, the third molar, that is already formed, so it's adult morphology. So again, it's very funny because a lot of people have preconceived ideas, including scientists, and myself included, because when I started working on this specimen, I was convinced it was Homo. Everybody thought it was Homo, so I said, okay, it must be Homo. But when I found the results of these analyses, started to question what we knew and what happened. Yeah.
Have you compared the morphological disparity within extant species?
Yes, not in this study,
and
but
within fossils?
Yeah.
And do you find maybe greater disparity in fossils, meaning that maybe in some cases you have actually several species and not only one? Could it be like some kind of clue that you know that in a living species there is not so much variance?
Mm-hmm.
If you quantify this variance, could it maybe inform you on
yes,
whether the sample that you think are the same species, actually it's not. It's too big.
So in this case, I didn't want to work at species level because for me, the species are not well defined, and it's really troublesome to work at species level because in some cases we have only 1 or 2 specimens per species. So it's not enough to do any stats. But if we work at genus level,
the genus level,
then we know there are different species. We cannot know exactly how many species, but we know there are different species. And you can see here that the degree of variation for each species is more or less the same. It seems that there is more variation for Sauropithecus, but you have to take into account that it's representing also a much longer chronological time period. Maybe here you have 2.5 million years of life of Australopithecus.
Possible to ponderate the disparity method?
Sure. Yeah. So I didn't do exactly what you suggest, but I did something similar, comparing the distances between specimens and the density plots, and basically all of these genera present more or less the same kind of variation, more or less. There is a bit more again in some cases, but it's very minor and it's not really statistically relevant. So the problem is also the differences in sample size. I have much less specimens for early humans, or Homo erectus agaster, than for the others. I'm starting to have a larger database, and it starts to be more homogeneous and more balanced for each sample. And it seems indeed that there are not so many differences in terms of variation inside each genus. So yeah, no, this is a good point.
It would be interesting to have a look at how this evolved through all the extinct species that we know, and then compare with the fossil record.
So there were some species on the evolution of teeth, and they showed that there is a neutral evolution. So basically, it's constant and it's homogeneous through time and space. In general, in hominins, there are some exceptions. The exceptions are in islands. When hominins evolved in islands, because of genetic drift, the genetic pool was so reduced that like for any other animal, weird things happen very quickly. But if a species evolved on the continent, then yeah.
So it's a Brownian evolution.
Yeah.
And also,
sure.
I wanted to do another comment on the few slides earlier when you shifted from landmark-based to this deformetrica methods.
Yep.
And you demonstrated that there was a better separation of the groups. I know that there is some possible bias when you're using high-dimensional data in between groups PCA or CVA.
That's
That when
what
you use like supervised classification, you are artificially increasing the separation between groups.
Yes and no.
So
Yes.
how do you account for that? And have you tried also to sort the individuals with non-supervised methods? I don't know, like neighbor joining or hierarchical classifiers,
Yep.
to see if you can go back on your feet and find the same genus classification.
Yeah, yeah. Um, so, for example, BGPCA, you can use a larger number of variables than individuals. It doesn't, I mean, mathematically it works. And I know some colleagues argue that it's not ideal, but canonical variates, you cannot do it. And for that reason, we use the PC scores. And so a selected number of PC scores, which are we give the best classification scores. And you can see here that the CVA based on a few PC scores and the BGPCA using all of the raw data give the same kind of plot and the same results. And also the hierarchical cluster, it's the same. So yes, we can account for these issues related to the higher number of variable. But also I've seen some colleagues using semi-landmarks and put a lot of semi-landmarks, like surface semi-landmarks and so on teeth or whatever. And sometimes it exceeds the number of 500. And since it's three-dimensional data, you have 1,500 data, and it's maybe comparable to that. So yeah, the issue is now the main issue I had when I tried to publish this paper is the people doing geometric morphometrics. They said it's not comparable. I mean, compared to landmarks, you are here using non-homologous comparisons because you don't have one point for one clear feature. But I could show that when I put landmarks and when I do the deformation analysis, I get the same kind of information. So basically it works, and I just get a better separation because I take into account probably more diagnostic features with the surface deformation. So almost done. So coming back to that, my goal is not just to say, okay, this is a different species or this is, whatever species it is, but these have very important implications in terms of biology and ecology. So for example, here you have this specimen. It was attributed to Homo, but in fact it's a Paranthropus. And you can see that compared with the earliest human, it's an outlier. If you remove it from humans, then it makes more sense. It's probably not the same species as that one, or we are not sure, but it's also probably not human. And this means that this individual didn't have the same behavior and didn't eat the same thing as humans. So the implications is if you can separate better humans, Australopithecus and Paranthropus, then you can better understand what they were eating, what they were doing, or they were living. And then, of course, you can better understand human evolution in general. So the same here, there are different individuals, and you see this fossil, it shows a very different signal from Paranthropus, Australopithecus, or other fossils attributed to the same genera. And for years, this has been attributed to humans, but in fact, it belongs to the new species I just mentioned, Paranthropus capensis, and you can see that probably this species, which is smaller jaw and smaller teeth, probably didn't eat the same thing as robustus species did. So again, if you can separate them morphologically, it has also implications in terms of biology and behavior and so on. And just very quickly, one last thing. I don't know if I have. Yeah, I still have a bit of time, I guess.
Yes.
Okay. Just one last topic. I also work in Asia, and in Asia, we now realize that there were many more species than we knew before, and especially that these species probably interacted with each other. And one of the most mysterious species in the last 10, 15 years is not yet described as a species yet. It's called Denisovans as a group. And they were found first in Denisova cave in the Altai in Russia here. And you can see here more or less all the fossils we know for Denisovans. They are the red ones here. And the other ones are Neanderthals. I will mention that just after. And these ones, we don't know what they are. And how do we know that these are Denisovans and these ones are Neanderthals? Based on phylogenetics. Because these fossils were found in Russia in a very cold environment. It was also in high altitude in mountains. It acts like a refrigerator and it could preserve DNA. So we knew the geneticists in 2010, they said, okay, we have 4 specimens, we could have DNA and we don't know what they are. They are not Neanderthals, they are not modern humans, probably not Erectus or other species, but we don't know what they are. So it was the first time that a species was somehow identified without even naming a real species name, based on genetics. And for a long time, only these fossils could be known as Denisovans, and nobody knew what they are or where they could be found elsewhere. Now you can see on the map, we have identified a number of sites where we can probably find Denisovans.
Okay, and to be clear, because those teeth are like 100,000 years old, and you say because they are in a refrigerator,
Yeah.
we could perform DNA analysis. So it means that in a typical, like in Paris, if you found a fossil, how far can you go until it's impossible to perform DNA analysis?
That's very valuable. To give you an idea, it depends on the climate and the environment, of course. In Laos, for example, where I will show you the example just after, or in tropical areas, I think the oldest DNA we have is around 3,000 to 5,000 years old.
Okay.
So we cannot retrieve DNA from very hot and humid conditions. It doesn't preserve, it's just destroyed. But if you go to the North Pole and, for example, I think in Greenland, they found DNA up to 2 million years. It's not human, it's plants and other animals, but still, it preserved. And in Europe, the oldest that was recovered so far is around 430,000 years old from Spain. It's in a cave that the temperature probably was very constant, very stable through time during all of these years. So it kept DNA preserved. So again, it depends really of the conditions of preservations. Sometimes you can have very old DNA, sometimes you don't find anything, even if it's recent. And you remember this one, Denisova 11, which is half red, half blue. So the geneticists, they were able to sample this small piece of bone. So it's just 2 centimeters. Morphologically, you cannot say even if it's human or not. But thanks to phylogenetics, they were able to say that it belonged to a human and probably first generation hybrid between Neanderthal mother and Denisovan father. And this individual lived around 90,000 years ago, at the time period where both groups could be found in the cave or near the cave of Denisova in the Altai. So that was really, really a huge discovery at that time. First of all, because we knew that our ancestors, different species could probably interbreed, but we were not sure. And that was the proof, the first genetic proof. But also because it's the first generation one. So it's super important for understanding the biology and the life of these 2 species at that time in this place.
So that means we have data of Neanderthal and Denisovian genetics.
Yeah.
Like genes and...
We do, yes. So for Neanderthals now, probably 30 genomes or 30 from 30 specimens we have genetic data. Denisovans, we have at least 4.5, maybe now 5 or 6. Some are not published, but not much.
5 or 6
genomes.
And so we could say that it is half.
Yeah, in this case, yes. Based on on the study of modern animals and what they know from hybridization between modern animals, they could show that it's the case of hybridization in human species.
How big is this cave?
It's
But
not
you
a
have to imagine, because you tell me there are Neanderthals, maybe another species, so it's like a really big cave?
No, it's not a very big cave, but they probably live there. There are also, some stone tools and other things, and we have traces of presence from maybe 140,000 or maybe a bit earlier to 40,000 years. So for nearly 100,000 years, they were occupying this cave, maybe not always at the same time, maybe not the two groups at the same time, but alternatively, and they came here. It's probably a good shelter, a good place to stay, I guess, and yeah.
Yeah, and because I listened to the interviews about the Grand Mandrins,
Oh, yes.
and because it's a really good cave right on the Rhône River Valley, so you can understand that people would settle there at every era. Basically, it's just a very good spot.
Yeah, I've never been to Denisova, so I cannot give you a lot of information on the context. But generally, the cave that were occupied, they were very strategic for hunting, to have good resources, whether it's water or good stone that then they can use to make tools or things like that. So they were generally on top of hills, mountains, or at least very protected and so on. So yeah. And thanks to genetics again, and including the analysis of modern people, modern populations, we know that, so first we know that Neanderthals and Denisovans exchange genes. We even have the evidence from a hybrid, but we know also that Neanderthals contributed to modern human populations in Eurasia, that Denisovans contributed to modern populations in Asia, mostly in Southern Asia and Oceania. And there is also evidence that an unknown species, we don't know what it is, some call it the super archaic, contributed to Denisovans just after they split as a species from Neanderthals. So thanks to genetics also, we know that Denisovans and Neanderthals, they are sister taxa, they are closely related, more closely related to each other than with us, modern humans, Homo sapiens. And in this context, we need to find more fossils, and with some colleagues, we have been excavating in Laos, in caves. This time it's a very small cave. It's probably 5 meters deep and 1 or 2 meters large. So it's not a place they occupied, they didn't live here. But probably the fossils that we found, they were trapped. It's kind of natural trap on top of the hill, and they fall down in the fractures and accumulated. And so you can see here some layers of soil, of sediment, and inside there are a lot of fossil teeth, lots of animal teeth, but also in 2018 we found this human tooth. And we analyzed it. We first dated both sediment and the animal teeth, and the chronology is roughly between 160 and 130,000 years old. But we had a question, what species it is? To which species does it belong? We didn't know. And just after we found the tooth, there was a publication of this mandible in Nature announcing that it's a Denisovan. Before that, we had no idea what the mandible molar of Denisovan looked like. And this was very fortunate because, as you can see, the first molar of this individual was very close to our specimen from Laos. And it's also very close to Neanderthals, which are here in red, and it's different from modern humans or from Erectus. One of the molars of this mandible, it's falling with Erectus, but it's an anomalous molar, and you can see here that it's much larger compared to the first molar, which is normally not the case in other species or in other individuals. So this one is a weird morphology, something unusual. But if you consider just the first molar, then you can see that it falls close or within the Neanderthal range. And it was attributed before to Denisovan based on molecular data. In this case, they didn't use DNA, they used proteins. But it works the same way. You can also separate species. And our tooth was closest to this specimen. So we found the Denisovan, but not in Russia, in Laos, in a tropical environment, in tropical forest, in a very hot climate, so very different from what we knew for Denisovans. So thanks to this discovery, and also we also use proteins, and we could say that it's a girl. Because the crown is not completely formed, there is no root, it's not broken, it's just not formed yet. So it means that it was a young individual, a young girl, probably around the age of 8 years old, if we compare with modern humans, what we know for modern humans. And this girl lived in a very different context from the other Denisovans we know from the Altai. So it also gave us the information that, like Neanderthals or like Homo erectus, Denisovans were very flexible species, capable to live both in the north, in the very cold climate, even in altitude, in the mountains, or also in the very tropical forest of Laos.
And just because maybe they lived at the time when Laos was a steppe tundra environment, or it has always been.
Thank you. That was the next point.
Okay.
So that's what we thought. And to be sure that it was living in the forest, because today it's a tropical forest, we did some geochemical analysis. And in fact, you're right, it was not really living in the forest. So thanks to the carbon isotopes, we can separate the individuals that live in close forests from the ones living in the more open environments. And this individual, this young girl, was fed on animal meat that came from more open environments. So probably they were living in a kind of steppic environment. But there were also some forest around, because all the animals from the same cave, they show that there is a lot of forest around. But the animals that were hunted and fed to the young girl probably came from the more open environments that was around the cave, probably in the valley. And just to give you a comparison, this is a specimen that is attributed to Homo sapiens, dating to almost 60,000 years old. So much younger than this specimen, but still quite old. In the same area, exactly 100 meters in a cave found in 100 meters from this one. And you have the same kind of animals living in the forest, also some living in more open environments. And in this case, Homo sapiens was really living in the forest and eating animals that lived in the forest.
And to be clear, like, does the isotope indicate like from which source they are drinking water, or like?
No. In this case,
it's forest versus steppe.
In this case, we also we know based on carbon isotopes, but we also know based on the species, because we know that some species live in the forest and some other species they live in open environments. So the combination of the information of molecular biochemical information and the biological or morphological information, we know that it fits. Yeah.
Okay. And maybe while we're at it, if you come back 2 slides earlier,
sure,
or even earlier, I don't understand why you have this kind of crust. They did not fall down, like in your.
Because that one was calcified. So that's a very common process in caves in Laos or in Southern Asia. The thing is that you have a lot of water, and with the monsoon and it's a tropical environment, there is a lot of water that goes into this karstic system, into the limestone. And in some cases, you have like huge floods that remove some parts of the sediment. But if some layers were calcified on top, it can happen when, like the stalactite, when you create a stalactite, you have calcite that goes down with the drops and make something solid. So you can have the same, that with just water flowing on top of the soil, it leaves some calcite, and it will harden the top part of the sediment. But below, it's softer, and if the water goes below at some point, then it will just remove everything below and leave just this part. It's very frequent. And sometimes, if there is a strong current and all the cave is filled with water, you remove everything and you cannot find anything. So we are also lucky that in this case, not everything was removed. And I think I'm done. Thank you very much.
Okay. Do you have any other question? Yes.
Just can you give again the reference of the methodology for making the graphs with Deformetrica?
Oh, yes. Yes, so, but there is not specific package. I mean, I'm just doing a PCA first. And then I'm using the PC scores for the canonical variate analysis, or just the raw data for the BGPCA, or you can use also the PC scores. It works also. All the PC scores for the BGPCA, for the between group PCA.
Can I have the paper that?
Sure. Every paper that I've published and showed you uses the same method. The last one was published in PNAS in 2022. The title is no. Well, I can send you the data if you want. I can give you, just give me your address, or I can give you my email, and I can send you the papers. But if you need some specific information, I can also send you a script in R or whatever. And
yes, no, exactly. I think we'll be happy to see that, because for us, all of our work is okay. What kind of model is implemented in Deformetrica? And since you said Deformetrica, we said, okay, but you have so many different methods.
Okay.
It's not clear to us what exactly you're doing.
Okay.
But we'll ask you.
No, of course. So I'm using the Atlas deformation, and then I use the momenta and the control points as raw data. And actually, there is a package by Jean Dumoncel to easily import all of this data in R. It's called R Tools for Deformetrica. And then from there, I'm just doing the basic things I just mentioned. So PCA and BGPCA and CVA.
Yeah, because with Deformetrica, you got a deformation, and then the PCA, you apply it on the deformation, or on...
I think on the shooting momenta.
Yeah, on the shooting momenta, yeah.
Okay.
And when you show the PCA axis, and you show some shapes in all your graphs, you had these two axes with shapes on. So these are created by shooting with Deformetrica.
Yeah, exactly. And actually, it's very nice because it fits exactly the morphology. I mean, it's normal, but also people who are not familiar with the approach, they can really see the same morphology. Like, this is the typical shape of Neanderthals or modern humans, and this is the typical shape of Erectus. So this is really nice also visually.
Okay, and maybe another question is, you didn't talk too much about isotopes. I don't know, but when I met people who work with like cattle from the Middle Ages, they tell me, we do isotopes on the teeth, and basically we're able to tell, from which source they were drinking water every month of their life, and so we can know, for instance, whether you had seasonal patterns.
So with strontium isotope.
Yeah, exactly. But you didn't talk about this kind of stuff, so I assume that it's not possible on the Swiss.
No, it is, but I just couldn't speak about everything we are doing. And also, this is not my work. I'm co-author on some of these studies, like this one for the isotopes of carbon. But this is also the work of other colleagues. And we work with someone called Renaud Johannes Boyot. He's in Australia. He's French, but he's in Australia. And he's doing all of the geochemical analysis for us. And he's doing a lot of things. He's also doing dating, but very interestingly, recently he developed this approach to map the chemical elements or isotopes. And we can also, you see some bendings here, and it corresponds to different kind of stages in the life of the individual. And you can see seasonal changes. So there are a lot of things that you can say based on this analysis, yes. I just couldn't show everything, but yes.
Okay, so probably we are good for the open session. And then, yes, Michael. Thank you.